Good morning, all of you. All of you, please settle down. We'll start with the lecture. Okay. Good morning. I'm Yasmeen Shaikh from Meetanagar School. Right. So we are going to continue with the lecture which we have started yesterday. Right. It was the 16th chapter of Science 2, that is heredity and variation. Okay. So I'll share your screen of presentation. Okay. So here it is. It is chapter 16th, heredity and variation. It's an important chapter. So we'll revise what we have studied yesterday. So in the previous lecture or yesterday, we have studied about heredity, right? What is heredity? So I've given you the example. The baby of the elephant is an elephant. The baby of monkey is like monkey. The baby of human being is like human being. So this is all because of heredity. The characters are transferred from one generation to another. That is also called as inheritance, right? So, and genetics, it is a subject which studies all these topics. Then also we have studied about inherited traits and expression of traits, how the characters are expressed, right, which are present on the chromosomes. We have studied what is chromosome and what are the different types of chromosome. I hope all of you remember, right? Chromosome is a genetic material, and depending on the presence of centromere, there are 4 types of chromosomes, right? Which are those? Do you remember? Metacentric, submetacentric, acrocentric, and telocentric, isn't it? Okay, so today we are going to start with the next point of the chapter, that is DNA, right? It is an important, very important part of the chapter, right? DNA, all of you must be knowing the full form of DNA is deoxyribonucleic acid, right? And it looks like this, right? I hope all of you know where this DNA is located. Yes, it is present in each and every cell of the body, right? It is present right from bacteria, viruses, plants, animals, human beings, in all the cells of all the living organisms, right? So, we are going to study the structure and the function of DNA. So, first, let us know this was the person, Fredric Mesher. So he was the first person to identify DNA or to find out DNA from the cell. So let us see how he has found. So he was working with white blood cells in 1869, right? So he was doing his work, he was doing the research on the white blood cells in 1869. So white blood cells are the major components of pus in the infection, right? When when your body is getting, when our body gets infected, so sometimes pus is there inside. So pus consists of white blood cells. So he has collected the pus from many hospitals, and he was doing research on that. So, as a result, Mesher collected a lot of pus from bandages at a local hospital. What he has done is added a weak alkaline solution to the white blood cells. Right? We use acidic and basic solutions. So he has taken from the laboratory some kind of alkaline solution, that is basic solution, and he has added to white blood cells. And when he did that, the cells decomposed, right, and caused the nuclei to move out of the solution, right? So what was his observation? As soon as he poured the alkaline solution on the cell, the cell decomposed, it bursted out, and the nuclei came outside the cell From the nuclei, Mesher isolated a substance and he called it as nuclein, right? And then from that nucleus, he has got a substance. He has found that there is some substance, so he named that substance as nuclein. After chemical analysis, nuclein was later renamed as DNA, right? So he was the first person. This is general knowledge, also, students. So you should know he is Fredric Mesher, who was the first person to find out DNA from the cell. Okay. Okay. As I've told you, this DNA, deoxyribonucleic acid, it is found in all the living organism. As soon as you say that the organism is living, it consists of DNA, right? So bacteria, virus, plant, animal, human beings, in all the organisms you will find the DNA. Okay, so this should be very clear to you. It is present in each and every cell of all the living organisms. So I've told you the location of the DNA. Where is the DNA located? You can observe the slide. The cell is shown to you. Inside the cell, there is a nucleus. We know that inside the cell there are different cell organelles, isn't it? You must be knowing, we have studied the cell organelles. So there are different cell organelles. For example, there is a nucleus, there is a endoplasmic reticulum, Golgi bodies, lysosomes, ribosomes, and many things are there, right? So, but the cell inside the nucleus, this DNA is present. Then also inside that nucleus also there is a structure called as chromosome, and in this chromosome, this DNA is present in the highly coiled form. Highly coiled form. I've told you yesterday the DNA is present in a very compactly coiled form. So if you will uncoil that structure, the DNA is very long, right? So and after uncoiling, you can see the DNA looks like this. Okay. So, uncoiling of the chromosome. You can imagine in a simple cell, what we say in a cell, so many things are going on. Now, these chromosomes, these are the coiled form of the DNA, and you can see if you uncoil this. Okay, so I'll show you with the help of a pointer. See now, the uncoiling is going on. When you uncoil this chromosome, then you get a such kind of structure. And if you go further, you can see that it consists of DNA and the histone proteins, right? This DNA molecule is coiled with the help of histone proteins. These red color balls, what you can see here, these are nothing but the histone proteins, right? So the DNA, it is coiled around the histone proteins, and then it is again coiled and coiled to form a supercoiled structure that is called as chromosome, right? So this is the basic information of DNA. DNA is deoxyribonucleic acid. Then chromosomes are mainly made up of DNA and proteins. It was discovered by a Swiss biochemist, Fredric Mesher, in 1869 while studying white blood cells. It is present in the nucleus, so called as nucleic acid. So I would like to elaborate this point. So previously it was thought that DNA is present only in the nucleus, and that is why it is called nucleic acid. But later it is also found that it is also present in some other organelles of the cells, right? It is also present in mitochondria, right? So it is present in other organelles also, but basically it is called as the nucleic acid. It is present in all the organisms from viruses and bacteria to human beings. It controls the functioning, growth, and division of the cell and are therefore called as master molecules, right? As we have brain in our body, our brain controls all the functions of our body. In the same way, this DNA controls all the functioning of the cell. So, all the functioning of the cell, as well as the growth of the cell and the division of the cell, everything is controlled by DNA. So, that is why it is called as the master molecules of the body. Right. So the question is asked in the exam sometimes. Describe the structure of DNA, right? So you need to write the information on DNA and explain the structure in details. Okay, so here are the scientists who have given the structure, who has found found the structure of the DNA. So in 1953, Watson and Crick produced a double helical model of the DNA molecule. Right? See, you can see here. These are Watson and Crick. Okay, so as per this model, now you can see this is this is the structure of DNA molecule, right? As per this model, two parallel threads of the nucleotides are coiled around each other, right? So I'll show you the parallel threads. What they're calling is, see this structure, this coiled structure. This is one strand, and here this is the other strand, right? So, as per this model, two parallel threads of the nucleotides are coiled around each other. This arrangement is called as the double helix. Right? This is called as the double helical structure. This structure can be compared with a coiled and flexible ladder. Right? So, if you see a ladder, it looks like a ladder, right? A coiled and flexible ladder. Each strand in the molecule of DNA is made up of many small molecules known as nucleotide. Right? So here you can see, so the nucleotides. Now, for example, now each strand is this. Each strand, what you can see, the help of this backbone also and the middle strand also. So that each strand is called as a nucleotide. Right? So that is called as nucleotide. So here you can see, now I'll explain you in the next slide. This is the sugar phosphate backbone. This backbone, this is called as this is made up of sugar and phosphate. So that is, this is called as sugar phosphate backbone. And this strands are made up of the base pairs, right? And I'll tell you about these nitrogenous bases. They are made up of nitrogenous bases. So let us study in details the molecular structure of the DNA, what it is composed of, what are the different molecules which make the DNA, right? So the basic molecular structure, right? You know that our whole body is made up of different molecules, isn't it? What we eat is also a molecule. For example, whatever we are eating in terms of, we ask in the class also, teacher, what we are eating, is it a carbohydrate or a protein? Isn't it? It is classified into different groups: carbohydrates, proteins, lipids. So the carbohydrates are made up of starch, proteins are made up of amino acids, lipids are made up of the glycogen, right? So fatty acids. So uh, that way means our whole body, whatever we eat, also all those things are or are in a molecular form. So in the same way, this DNA is also made up of different chemicals, or what you can say, different molecules, different elements. Right. So we are going to study in details how it is formed. Okay. So, in the formation of the DNA, actually, the structure of the DNA molecule is same in all the organisms, right? This double helical structure is same for all the organisms. Each strand in the molecule of DNA is made up of many small molecules known as nucleotides. There are four types of nitrogenous bases. Those are called as adenine, guanine, cytosine, and thymine. Okay, adenine and guanine are called as purines, and cytosine and thymine are called as pyrimidines. Right? So these are the nitrogenous bases. Actually, it is in short, it is called as ATGC. A always combines with T, that is AT, adenine and thymine, and guanine always combines with cytosine. So this always the combination is the same, AT and GC, because purines always combine with pyrimidines, and purines cannot combine with purines. Pyrimidines cannot combine with pyrimidines. So there are four types that you have to, I'll again revise so that you remember these words. These are important words. There are four types of nitrogenous bases. It is adenine, guanine, cytosine, and thymine. Adenine and guanine are purines, while cytosine and thymine are called pyrimidines. In the structure of a nucleotide, a molecule of the nitrogenous base and phosphoric acid are each joined to the molecule of sugar. As there are four types of nitrogenous bases, nucleotides are also of four types, right? So, in the structure of the nucleotide, what all things are present? A nitrogenous base is there, phosphoric acid is there, and the molecule of sugar is there. So, the molecule of sugar and phosphoric acid, it forms the backbone and it is combined with the nitrogenous base, right? So, in this way, the structure is there. And here it is written, you can see down. Adenine always pairs with thymine and cytosine always pairs with guanine. Right? So this is we say this is nothing but the uh, I'll tell you in the next slides. It is also the codes, right? These are also the genes. These are called as the genes, and these are the coding for certain proteins, right? So, for example, if the combination is AT, right? So, AT, ATGC. So, three genes code for one protein, right? So, if the combination is AT, ATGC, then this three genes or there's three codes, that is three nitrogenous bases, it gives information to the cell. It is a gene, and a certain kind of protein is it is asking for the synthesis of certain kind of proteins, right? In this way, there are, you can make millions of combinations of all these only four nitrogenous bases, ATGC. You can combine in a different way. Many different combinations are possible, and which are nothing but the genes, and which gives the information about the synthesis of different proteins, right? It can be AT, right? It can be AT, ATGC, it can be GC, AT, GC, it can be AT, GC, AT. So that way, many different combinations are there. If you write on your own, you can find there are thousands or millions of combinations you can make out of that, right? So the studies are also going on in order to find this, that these the pairing of nitrogenous bases gives the synthesis of which type of protein, right? So that also research is going on. So here you can see this is the molecular structure as you have been taught in the schools, isn't it? How to write the sugar molecule, right? So what is the simple molecular formula of sugar? You know, C6H12O6, right? So that way, here, so here you can see this is the sugar molecule. We draw the sugar structure like this, right? So here you can see this, and on CH2 carbon, on this carbon, phosphoric group is attached. This is called as a phosphoric group, phosphoric acid, right? And here on the other end, on this hydrogen, the nitrogenous base is attached. This is the structure of adenine. As you go in the further classes, in 11th and 12th standard, you will be taught to draw the structure of the molecules, right? So, but for now, you just remember this is a sugar molecule, this is phosphoric acid molecule, and this is a nitrogenous base, adenine. So, this is how the arrangement of the DNA is there, right? Though we say that it is a thread-like structure, it is highly coiled structure, but it is the molecular. This is the molecular structure. Sugar phosphate backbone is there attached to the nitrogenous base. You remember this. There is a sugar phosphate backbone and attached to the nitrogenous base. Okay. So here you can see the detailed structure. Now here, see, this is called as the cell nucleotide, right? Can you see? From here, this is a sugar molecule. This is phosphoric acid. This is nitrogen base. This is of one strand. This is of the one strand. This is one strand of the DNA molecule. This is the other strand of the DNA molecule. They go anti-parallel. If it is moving in this direction, yeah, the other one will move in the other direction, right? So this is sugar molecule, this is phosphoric acid, and this is the nitrogenous base of one strand. And here's the other strand. This is sugar molecule, this is the phosphoric acid, and this is the nitrogenous base. Now see here, you can see the combination TAGCCAT. So in this way, and these two nitrogenous bases, they are together by a hydrogen bond, right? These purines pyrimidines are joined by hydrogen bond. So this is what is a molecular structure, right? So also there is 3' end and 5' end of the DNA molecule, but that is not given in our textbooks. That is not for our studies. It is when we do higher studies on this. So more detailed structure is there. Okay. So this is the one strand, and this is the other strand of DNA. That was about the molecular structure. So you should know the molecular structure of DNA also. Okay, so students, I hope you have followed this much. You have followed who found out the DNA and what is the full form of DNA, where it is located, and also you should have understood the detailed structure of the DNA and the molecular structure of the DNA. Okay. Now the next point is gene. What is gene? Gene is nothing but a segment of the DNA molecule, right? I've told you maybe three nucleotides together, that is called as a gene, right? So segment, a part of the DNA molecule, segment of the DNA molecule are called as genes. Due to variety in the sequence of the nucleotides, different kinds of genes are formed. These genes are arranged in a line. Genes control the structure and the function of the cells and of the body. Also, they transmit the hereditary characteristic from parents to offsprings, right? So, these genes, these are transferred from one generation to another by the process of cell division, and that is called as meiosis, right? And genes also produce different types of proteins which help in the growth of the cell as well as growth of the body. So a gene is the structural, what you can say, gene is the structural and the functional unit of life. Each chromosome, I'll again try to explain you where the gene is present. Each chromosome is made up of single DNA molecule, right? And the segments of the DNA molecules are called as genes. Due to variety in the sequence of the nucleotide, different kinds of genes are formed, and genes control the structure and the function of the body and the cell. Also, they transmit heredity information. That is why many similarities are seen between the parents and their offsprings. Information about protein synthesis is also stored in the genes, right? Which protein has to be synthesized in the cell. For example, a child is small and now he is growing taller in height, or the boy is growing, so this growth hormone will be secreted. So the gene will be stimulated for the growth hormone, and the growth hormone will be secreted by the growth hormone kind of this kind of, and protein will be synthesized in the body by all the cells, and finally, or with a result, the child will grow in height, right? So this was just an example of the protein synthesis for the growth of the body. So in this way, for each and everything, this genes synthesize proteins, right? So hence they are said to be the functional unit of heredity, right? So in the germ cells, we have said that the same kind of DNA is present in the autosomes, but in the sex chromosomes. So we have seen that in the sex chromosomes, half DNA, that half DNA of the male and half DNA of the female, it combines, and because of this combination, a different kind of human being, that is a child, is born with some similarities and some differences in his body, right? So your gene is also said to be the functional unit of heredity. So what kind of genes has been inherited, that will result in the new individual. Okay, so the next topic in our textbooks are Human Genome Project, right? Now, with the name itself, you can find out the sequencing of the genes on the DNA. Can you imagine the sequencing of the genes, whichever are present on the DNA? That sequencing has been done, and that is called as the human genome. So let us study in details what was this Human Genome Project. So, actually, in 1990, the Human Genome Project was together undertaken by all the geneticists of the world. Right? In June 2000, scientists of this project and Celera Genomics Corporation, it is a private industry in USA, right? They collectively announced the discovery of the complete DNA sequence of the human genome. Depending upon the findings of this project, scientists confirmed that the number of genes in the human genome is about 20,000 to 30,000. How many genes are present in the human genome? 20,000 to 30,000 different types of genes are present. Later, scientists discovered the genomic sequence of many organisms. So, after human genome, they have also found out the sequence of different organisms, including, see here, it is written, including beaver's yeast, the roundworm, and the fruit fly, right? Beaver's yeast, roundworm, fruit fly, we were studying human genome, right? We'll continue. So, what is the use of this genomic study? So, if we have studied the genome, so disease-causing genes can be identified, right? So, the hereditary, sometimes the diseases are hereditary, isn't it? We have heard that diabetes is a hereditary disease. So, the genes which are responsible for causing this diabetes. So, if we will study this properly, we'll study the genes properly, then we can also find out some cure about it. We can do the research for finding the uh, medicines or some kind of curing for such kind of diseases, right? So, if disease causing genes are identified, genetic diseases can be diagnosed and it can be properly treated, right? So, large use can will be there in future. So, more research is going on. The scientists are studying the different genes of the human beings and how, and also the disease causing genes, and they are working on it, that how this disease causing genes can be replaced or can be treated.
हेलो टीचर, सॉरी टू इंटरप्ट यू बट योर स्क्रीन इज नॉट. हाँ यस टीचर, अभी दिख रही है.
Okay, so I hope all of you can see the screen now. So the next is DNA fingerprinting. Now this is also one use of the findings of DNA. The sequence of the genes in the DNA of a person, that is the genome of the person, is identified. You know, this sequencing of genes on the DNA is very specific. It is specific for a specific person. It is not the same. The DNA, the sequencing of genes on the DNA is the sequencing of the genes in the DNA of a person is different for different people, right? So it is not, it is unique, it is not the same for anyone. So that is why it is of much use. So it is useful to identify the lineage, right? And it is also useful to identify criminals because it is unique to every person, right? You must have seen, if you're watching the shows, you must have seen in CID or maybe in the different crime branches or in the crime patrol serial, you will be seeing that they are sending for any criminals. If they want to find out the criminals, they are sending it for DNA testing, right? So as the DNA is very specific, it is very unique for the person. It is not same, so that is why then they are. It is helpful for them to identify the criminal, right? Okay, so here it is. So we have finished with today's part. So I would like to revise what we have studied. So we'll quickly revise all the things. So today we have started with the with DNA molecules, right? DNA is the deoxyribonucleic acid, which is present in the cell, and these are the master molecules. So he was the person to first identify DNA, Fredric Redrich, and we have seen that DNA is present in all the organisms, right from bacteria, viruses, plants, animals, human beings. Also, we have studied location of the DNA. Those DNAs are present in the cell as well as in some other organelles of the cell. DNA is present in the nucleus as well as on some of the other organelles of the cell. And coiling of the DNA. For coiling the DNA is coiled around the histone proteins, which is coiled again and again to form a coiled structure, and then it finally forms a chromosome. Okay. Then your study started with the structure of the DNA. So it was given by Watson and Crick, and the structure is called as the double helical structure of DNA. Right. So we have seen the components of the DNA molecule. It consists of sorry. So sugar, phosphoric acid, and the nitrogenous bases are there. Sugar and phosphate it forms the backbone, while the nitrogenous bases makes the strand. So the combination of sugar, phosphate, and sugar, phosphoric acid, and nitrogenous base forms a nucleotide that is called as a single nucleotide. So the DNA molecule is made up of many different nucleotides. So here the four types of nitrogenous bases are there: adenine, guanine, cytosine, and thymine. So these are the molecular structure. This is the molecular structure you can draw, and you should try to draw and practice it. Then we have also studied gene. Gene is nothing but the segment of a DNA or the part of a DNA is called as gene. And these are the structural and the functional unit of heredity. It, these genes are transferred from one generation to the next, which is responsible for the similar and the variations of the organism. Human Genome Project, that is studying the sequencing of the genome or sequencing of the genes on the DNA. It was successfully completed, and now also the research is going on the disease causing genes and the treatment related to it. So this DNA sequencing is used and to identify the criminals. Okay. So before we finish, I would like to tell you students. So you do a small homework with the help of net. You everyone must be having internet. With the help of internet, all of you make a project that all of you make the structure of DNA, right? So prepare a model of the DNA and give a presentation based on it. So such kind of activity is also there in our textbook on page 193. You should draw the model of the DNA. You should make a model of the DNA, sorry. So you can do it with the help of the internet. So try to make the model. You will find it interesting. So I'll stop here today. So if you're having any doubts, you can ask me, students.
Yes, teacher. Thank you. Thank you so much.